All right, welcome back, folks. In this Edpuzzle lecture, we're going to go over DNA replication. We're going to go over it a little simpler than usual because we're kind of short on time this year. But anyways, here's our daily objectives. First, you should be able to generally describe the process of DNA replication. Remember, replication is when DNA makes a copy of itself. It makes an exact copy of itself. It needs that in order. If you have one cell that's going to divide into two cells, each of those new cells are going to need their full DNA. They're going to need all the DNA. So you need to make a copy of the DNA before the cell can divide. And for you regular bios, you should be able to identify at least 2 enzymes which are involved in the process. Now, honors bio, you should be able to identify at least 4 and maybe 5 different enzymes involved in the process. Okay, so like I said in the previous slide, the DNA replication is the process where a cell makes a copy or replicates its DNA. Remember, if you remember from cell reproduction, this happens during the S phase of interphase during the cell cycle. So it happens before PMAT, prophase, metaphase, anaphase, telophase. It happens before mitosis. It is necessary because when the cells reproduce or divide into 2 cells, both cells are going to need a complete copy of the DNA. So before they divide, the cell needs to replicate or copy its DNA. There are many enzymes required to replicate the DNA. And remember, enzymes are proteins in the body which speed up chemical reactions. I know it's been a long time since we did biochemistry back in the fall, but enzymes are proteins which speed up chemical reactions in the body. All right, so here's step 1 in DNA replication. If you remember, DNA is called a double helix because it looks like a twisted ladder. So up here, it still looks like a twisted ladder, but in step 1, the DNA molecule is unzipped by the enzyme Helicase. So that's one enzyme you're going to need to know, Helicase. Now, Helicase is not really shown here, so I'm going to draw them in. It'd be right here. This is the enzyme, this little red thing I'm drawing. And it is moving this way, and it is unzipping the DNA molecule. So right here, you can see that thymine and adenine are still bonded together. But when Helicase reaches there, it is going to unzip it just like when you pull on the little handle in your zipper to unzip your sweatshirt or whatever. It's going to unzip. So like these would be like little nitrogen bases that are still bonded together. But once this little handle of the zipper reaches them, it's going to unzip them. It's going to separate them just like this. And that what makes what's called a replication fork. And all the replication fork is just unattached bases. So now that Helicase went through and unzipped this DNA molecule, now this thymine right here, it's no longer bonded to this adenine. This guanine, the G, is no longer bonded to the cytosine, the C. So it's leaving these exposed bases where now another base can be added to each side. And that's what's going to happen in the next step. Okay, so step 2 of DNA replication, you have free-floating nucleotides. And those are these guys. Here's a cytosine that's just floating around the nucleus. Here's a guanine. Here's a thymine T. Here's an adenine A. Now, this enzyme called Polymerase is going to attach the free-floating nucleotides to its complementary base. So, for example, let's say this is polymerase. It's going to come and attach an adenine there. For this G right here, it's going to come and attach a cytosine. For this adenine, it's going to come and attach a thymine or T. Remember, it's according to the base pairing rules. A with T, C with G. So this G right here, it's going to come and attach a C. With this G, it's going to come and attach a C. Same thing on this side. For the A, polymerase is going to come and attach a C. For the C, it's going to come and attach a G. The T, it's going to come and attach an A. So you can see we started with one single DNA molecule, and now we're actually making one molecule of DNA over here, double-sided, and we're making a second double-sided molecule of DNA over here. So that's all DNA replication is. It is first unzipping the DNA molecule by helicase, unzipping it, and then it's just adding the other part of the zipper. So this cytosine right here, it's going to add a guanine, a G. And it does this by the enzyme polymerase simply attaching these free-floating nucleotides, which are floating around the nucleus, and bonding them to its complementary base, which means it's bonding adenines with thymines, and it's bonding cytosines with guanines. Now, if the DNA polymerase makes a mistake, these are extremely rare, but those are what's called mutations. And mutations is what can lead to things like cancer, but it can also lead to beneficial things, and you'll learn about that when we get to evolution. All right, so another key term. DNA replication is said to be what's called semi-conservative because one half of the new strands are new and one half are old or conserved. What that means is, remember, the original DNA molecule was unzipped by the enzyme helicase. So this strand of DNA out here, A, and this strand out here, D, those are the old strands that was left over from the original DNA strand that was unzipped by helicase. Now, these middle strands, this B and the C, which are being built right now, like right here, this T is going to get an A bonded to it, and this G is going to get a C. Well, these blue strands, these are brand new. So half of the new DNA molecules are brand new, and half of the new DNA molecules are old. They were conserved from the original DNA molecule. All right, so now a couple of advanced concepts when it comes to DNA replication. A lot of you regular bio and honors bio students are amazing students, and obviously you're going to college. And if I didn't challenge you, I wouldn't be doing my job. So here's some advanced stuff. Most of this is extra credit, so take notes if you want extra credit. All right, so DNA has what's called a 3' and a 5' end. Now, all that means is if it's a 5' end, you can see here, it ends with the phosphate. Now, hopefully you guys know what a DNA nucleotide is. Remember, you got the phosphate bonded to the 5-carbon sugar called deoxyribose, which is bonded to one of 4 different nitrogen bases. In this case, it's thymine or T. So if it ends with a phosphate, it's the 5' end. If it ends with a deoxyribose sugar, it's the 3' end. And it's always opposite. So if it's 5 up here, it's going to be 3 down here. If it's 3 over here, it's going to be 5 over here. And if it's 5 over there, it's going to be 3 up here. So it's always opposites. Now, the only reason this really matters is because DNA polymerase, the enzyme, can only add new nucleotides in the 5' to 3' direction. So this DNA molecule over here has already been unzipped by the enzyme helicase. And I'll draw helicase in over here. So helicase is going this way and unzipping this DNA molecule. Well, you can see here is the 3'. So this side right here that it's going to add, this is going to be the 5'. So on this side, DNA polymerase can just start adding bases. T, it'll put an A. A, it'll put a T. C, it'll put a G. A, it'll put a T, and so on. So in this side, it can just keep following behind helicase as it unzips and just keep following it all the way down. And that's called the leading strand. So this strand over here would be called the leading strand. Now, on this strand over here, it has to go the opposite direction. So on this side, it's going to have to go down because it can't go, because if it goes up, it's going to be going from 3 to 5, but it can only go from 5 prime to 3 prime. So on this side, it's going to have to go down. So this T, it's going to put an A. The G, it'll put a C. The T, it'll put an A. And the A, it'll put a T. So it has to add the nucleotides in little tiny chunks, and those chunks are called Okazaki fragments. Okazaki fragments. So as helicase keeps moving along and unzipping it, polymerase comes and adds the bases, but it's got to go the opposite way. So it can't just follow the little unzipper, it's got to go the opposite way. With G, it'll put a C. With C, it'll put a G. So on the leading strand, DNA polymerase can continuously just follow helicase and without stopping just keep adding bases. G, it'll put a C. C, it'll put a G. T, it'll put an A. Never has to stop. C. But on the lagging strand, it has to keep putting these bases, these nucleotides on in little short chunks called Okazaki fragments. And so the leading strand is this way, where it never has to stop, where on this side where it has to put them together in chunks, that's called the lagging strand. Now once again, this is extra credit, so don't freak out too much if you don't understand it. And here's a little diagram that basically shows the same thing. So you see here's the 3' end, so that would not have a phosphate on the end, so this is 5'. So you can see DNA polymerase here. It's just following helicase. Helicase is right here and it's unzipping this DNA molecule. And here polymerase can just continuously follow behind helicase and just without stopping just keeps adding these free-floating nucleotides to the exposed bases. But on the other side, it cannot follow helicase because it'd be going 3 to 5'. So it has to go in the opposite way. So once helicase unzips a little segment of DNA, polymerase adds little chunks, and they're called Okazaki fragments. So the nucleotides have to be added in little chunks called Okazaki fragments. Now, if you'd like a little more extra credit, there's another enzyme called Primase, and its job is it lays down what's called a primer. And basically, the primer is like a little starting block where polymerase can bond onto and begin adding the free-floating nucleotides to the exposed bases to add the other side of the DNA molecule. Now, if you want a little more extra credit, there's another enzyme called Ligase. And you can see right here, in between the Okazaki fragments, those nucleotides are not bonded together. So ligase comes in there and actually bonds those Okazaki fragments together. It bonds them together. And even more extra credit, I don't even think I mentioned this one, but DNA polymerase 1, this guy, the little red primers, the little starting spots for DNA polymerase, well, DNA polymerase 1, it comes in and replaces the red RNA primers and replaces them with DNA nucleotides. Once again, that is extra credit, so if you don't understand it, that's fine. So polymerase 1 will also come over here and replace this primer with DNA nucleotides. Okay, last slide. So remember, in our daily objective, you had to know 2 enzymes, and here's the 2. Helicase. That's the guy, the little thing, if you hold it and you unzip your sweater, that's the one that breaks the hydrogen bonds between the bases to unzip the DNA molecule. The other one you need to know is DNA polymerase. And precisely, it's DNA polymerase number 3, and that's the guy that's going to add the other side of the zipper. It's going to attach free-floating nucleotides to the exposed bases, which basically means it's going to add A's to the T's and T's to the A's, and C's to the G's. And remember, it works in the 5 to 3' direction. Don't need to remember that, but just in case you want to. For extra credit, remember Primase lays down the primer, which is a starting spot where polymerase can begin to lay down nucleotides. And if you want more extra credit, ligase is the one that comes together and bonds together the Okazaki fragments. All right, this was a pretty complicated little Ed puzzle, but let's see how you do on your little quiz.